The world is one big biosphere. It is filled with biodiversity. The biosphere is split up into many different biomes. A biome is ecosystems within areas of similar climate and similar organisms. An ecosystem is a biological community of interacting organisms and their physical environment. For an example, in this image represents an ecosystem. The different animals interact with each other for food. In an ecosystem, there is a community that consists of a group of organisms or population of different species occupying a particular area, usually interacting with each other and their environment. In this image, there are species of turtles, species of fish, species of deer, and many more. An ecosystem has different species that are made up of many organisms. The species are all affected by the abiotic factors. Abiotic factors are parts of the environment that are non-living. Some examples are sun, wind, and water. The species are also affected by the biotic factors. Biotic factors are all the living things in the ecosystem. A great way to show how energy transfers through the ecosystem from one type of organism to another is a trophic pyramid. An ecosystem is made up of producers, for example, this apple tree and the grass, and consumers, for example, the bunny and the horse. Through each level of the pyramid, energy is being transferred. The arrows in this trophic pyramid represent the energy being transferred. Only about 10% of the energy is being transferred from one level to the next. The other 90% is lost through the metabolic process as heat and also by kinetic energy and even potential energy. First, the trophic pyramid starts with the sun. The sun gives off solar energy to the first level of the pyramid. The first level is known as autotrophs or producers, who make their own food through photosynthesis or cellular respiration. Examples are bacteria, algae, and plants. The next levels are the consumers. They are broken down into primary consumers, secondary consumers, and tertiary consumers. The primary consumers are herbivores. They consume the autotrophs. The secondary consumers are the carnivores, who consume the herbivores. The tertiary consumers are carnivores who eat other carnivores. Then, not shown in the inside of the pyramid, levels come scavengers, detritivores, and decomposers. Sometimes it is hard to understand the difference between the three. To start off, scavengers break up larger dead animals, or plants. Then, detritivores also eat dead animals or plants. However, scavengers eat on a bigger scale of the animals, and detritivores eat crumbs and smaller pieces of the leftovers that scavengers did not eat. For example, they would eat the leftovers and pieces of these animals in the pictures. All of the leftover nutrients and materials need to be recycled back into the ecosystem. This is where the decomposers come in. Decomposers recycle all the nutrients by absorbing the waste from the detritivores and the scavengers, and breaking it down into similar inorganic raw materials. Examples of decomposers are fungi, worms, and mold. The raw materials are all recycled and the whole cycle continues. So now we're going to be testing if there's ammonia, nitrates, and nitrites. If there are high levels, that means something is being decomposed. When an organism decomposes, the protein breaks down to leave nitrogen, and nitrogen in the form of ammonia or nitrate or nitrite. We'll be testing if a chicken decomposes in the soil. First, we must check the baseline, meaning the amount of ammonia, nitrates, and nitrites in the normal soil. First, I'm taking the soil and filling it up to 200. Then I fill the soil with water to 650. The soil is not fully dissolved into the water, so I have to go through a process of draining it and leaving the liquid top surface in order to get the successful results I want. I keep draining it using sponges and paper towels. What worked best is paper towels at each funnel, 3 funnels going into 1 beaker. You know it is working when the funnels get clogged and the liquid is clear. First, I am testing the baseline ammonia. I fill the soil water to line in a small test tube given in the ammonia kit. I then take the bottle 1 and add 8 drops. Then I take bottle 2 and add 8 drops. I seal the test tube and shake it. I wait 5 minutes for the test tube to settle in.
5 minutes later.
After 5 minutes, I compare the test tube to the fresh water color card. The results are that it is between 2 milligrams and 4 milligrams of the color card of the ammonia in the normal soil. Now I'm using a special soil kit to test the nitrogen. The nitrogen test will tell me the baseline for the amount of ammonia, nitrates, and nitrites in normal soil. First, I fill the cup with 100 milliliters of normal soil. Then I fill the cup to 500 milliliters, so they are 1 to 5 proportion. I combine both beakers into one large beaker. I stir the solution and wait 15 minutes to let some of the soil drain and dissolve in the water.
15 minutes later.
Then I take the container the kit gave me. The container has two sides, one test side and one side where I won't be putting chemicals in. Using the pipette, I put the solution in both sides up to the line shown. Then I take the substances in the small plastic pills that match the color of the container. I take the purple pill, which contains the chemical to test for nitrogen, and put it into the clear testing side. It is important to make sure that you do not get any in the other side. Then I seal the container tightly. I shake it for 5 seconds. Then I wait 10 minutes for it to settle. After 10 minutes, I compare the test side to the other side that has a color card chart telling me how much nitrogen is in the solution. The solution matches up to in between nitrogen 0, depleted, and nitrogen 1, deficient. Now I'll be testing if the chicken leg decomposed into ammonia, nitrates, and nitrites that is absorbed into the soil. First, you will need 300 milliliters of the soil and 600 milliliters of the water. Then I combine the two into one beaker. Then I stir and wait a half hour. I use the centrifuge to successfully dissolve the soil into the water. First, I put the same amount of solution into the beakers and place them into the holes of the machine. I press the on button and wait a few minutes for it to spin. After a few minutes, I take the beakers out and put the solution into a new beaker. Notice how the solution is very clear. This means that the soil is soluble in water. Now it is time for testing. The testing is the exact same procedure as it was for the baseline. First, I start with ammonia and put the chemicals in the test tube and shake. I wait 5 minutes for the chemicals to settle. The final results were that the soil had 8 milligrams of ammonia. The baseline was between 2 and 4 milligrams. This is a picture side by side. Then I test for nitrogen. I do the same procedure as before. I let it sit for 10 minutes. The results are N4 surplus. The baseline was between N0 and N1. Here's a picture side by side. This highlighted that the chicken leg decomposed into nitrogens absorbed into the soil.